land together. So this week we have Dr. Cosima Porteus. I hope I'm pronouncing that right. Uh, she received her BSc and MSc from the University of Guelph in Canada, majoring in marine and freshwater biology. Early during her undergraduate degree, Cosima became involved in research on freshwater mussels, fish, and salamanders, and became fascinated by how aquatic animals cope with various environmental challenges. She then moved to the University of British Columbia, where she obtained her PhD studying how fish sense and respond to low oxygen. As a postdoc at the University of Exeter, Cosima determined why fish exposed to ocean acidification lose their ability to respond appropriately due to olfactory cues that are vital for their survival in the wild. Her postdoc work was funded through fellowships from the Royal Society and NSERC, and she is currently an assistant professor at the University of Toronto, Scarborough, where her lab is continuing to study the effects and physiological mechanisms of anthropogenic stressors on aquatic animals. In 2023, she was awarded the Society of Experimental Biology President's Medal for the animal section for her contributions to this field of research. So without further ado, take it away.
Thank you so much. And thank you for the invitation and the chance to talk to you about my research. I want to start with just acknowledging the land on which I do my work, which is the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and I'm grateful to be able to work and learn on this land. I also like to acknowledge the land of the Huayat First Nations on which we conduct our research on Vancouver Island. And I'll be presenting some of that work later on in the talk. But I'm just kind of in my own journey towards truth and reconciliation, and I'm learning a lot about the kind of untaught history, or the history that is not taught in schools or I've ever learned about. So I'm kind of on this journey and learning a lot myself. And I think that one of the things that strikes me from many Indigenous cultures is some of the values in the way they treat the environment, in terms of treating not just the plants and animals as equals, but also the earth, the water, the air. And this is kind of very relevant to this talk and to my work in general, because I think if we as Western scientists would have viewed things this way, we probably wouldn't have been in the kind of climate change mess we are at the moment. It probably would mean that I would be studying something else, but I just want to acknowledge that we probably need to start to work towards kind of changing the way we view our environment. And with that, I'm going to start to tell you about some of the environmental mess we find ourselves into. And if you're not familiar with the term, the deadly trio of the aquatic environments, these are the three stressors or environmental challenges that aquatic animals usually are faced with. And they're faced with many others, like pollutants, but probably the three abiotic factors that are changing most quickly are those of temperature, low oxygen or hypoxia, and ocean acidification. In my lab, we study how these affect the sensory systems of animals, and I'll give you a little bit of introduction to how the animals that we study, how they sense their environment. But to begin with, I just want to say that I'm not going to spend too much time on temperature, or we haven't actually done too much work around that space. But I will tell you more about the effects of ocean acidification, And this has kind of been well studied, particularly by myself and a couple of other groups. And then towards the end, I'll tell you a little bit about low oxygen and hypoxia. I'm going to start by kind of going through what ocean acidification means and what it is, a little bit of chemistry, but not too much, but just to make sure everyone's on the same page. So I think everyone's aware that CO2 is increasing in our atmosphere as shown here. We've reached a level of currently of around 425 parts per million in our atmosphere. The CO2 in our atmosphere equilibrates with and dissolves into the top layers of the ocean, and not just the ocean, but any body of water or lakes or rivers as well. The CO2 reacts, most of it reacts with water, gives rise to bicarbonate and hydrogen ions. Some of these hydrogen ions do react with some of the carbonate present, particularly in the ocean, creating more bicarbonate. In the process, there's release of hydrogen ions, and this causes a decrease in pH. In orange here, what is shown is this increase in the CO2 partial pressure that we see in the ocean, and it's increasing at a similar rate to that in the atmosphere. Now, because of ocean currents, these high CO2 waters are mixed, and eventually the CO2 in the whole ocean rises, and therefore the pH is dropping. It is estimated that by the end of the century, the CO2 in our oceans will reach somewhere between 800 and 1,000 microatmospheres. So slightly different. We use a slightly different unit for that, but it's pretty much equivalent to parts per million. And then it's predicted that the pH could potentially drop as much as 0.3 to 0.5 pH units by the end of the century. This is on a business as usual model. So if we keep emitting CO2 at the current rates that we are currently emitting, and we're not doing anything about it, the pH in our oceans is going to decrease by this amount. You might not think that this is a large decrease, but I just want to remind you that the pH scale is logarithmic. Therefore, this represents a 120% increase in hydrogen ion concentration in just, 75 years, which is a lot considering the size of the ocean. Now, in my lab, and I'll get to this a little bit later in the talk, we are also concerned with another stressor, which is hypoxia. It's generally defined as low oxygen that cannot sustain life. We kind of think of it as oxygen that is lower than fully saturated water in aquatic environments, and I'll tell you a little bit why later. But overall, research is showing that there's an increase in severity and frequency of hypoxic events in aquatic environments. These are mostly showing coastal areas, but this is happening again in our lakes, in our rivers, even, and this is due to eutrophication, which is where nutrients from things like fertilizers end up in our waters. This creates additional nutrients, allowing algae to grow. And when the algae then die, they decompose, and the process of decomposition uses up oxygen from the water. This often starves aquatic animals of the oxygen and can cause massive die-offs, such as the one shown in this picture here. Now, what we study in my lab is how animals kind of detect their environment. We mostly focus on olfaction, and that is because many aquatic animals use it to find food, to find suitable spawning grounds, such as these salmon here. Apologies about the quality of the picture. It was actually taken just down in a nearby stream. Aquatic animals also use their sense of smell to avoid predators. We know that alarm cue, for example, is very important odorant that animals use to get away from a predator. They also use it to find mates, and really interestingly to recognize conspecifics or heterospecifics. So it's amazing that, for example, a fish can tell another fish is a sibling simply using their sense of smell. This is kind of one thing that's kind of always fascinated me. Now, a little bit about why we study olfaction. It's because out of all the senses, olfaction can be used over kind of the longest distance, because odorants are released into the water. They can be carried by currents over distances of 2 kilometers or more, compared to other senses such as their vision, which is often impaired, especially if the water is turbid. This can only used over a distance of maximum a few tens or 100 meters. What's also important to keep in mind that odorants are found in low concentration in the environment. So, for example, amino acids are found in the 10 to the negative 8 to 10 to the negative 6 molar, which is near their detection thresholds in many aquatic species. I've started being interested in how ocean acidification affects aquatic animals due to some early work that came out that mostly we're finding that exposing fish and invertebrates to ocean acidification was changing their behavior to and how they were kind of responding to these odorants. So since then, there's been dozens, if probably now over 100 studies looking at how ocean acidification affects the behavior of aquatic animals. And this is anywhere from freshwater fishes to invertebrates, sharks, you name it. What my interest as a physiologist was to answer the question, well, what is the mechanism by which ocean acidification has this effect? But before I go on to tell you a little bit about some of my work, I just want to acknowledge that there's a lot of controversy in this field, particularly surrounding this idea that whether ocean acidification does or does not affect the behavior, and particularly on coral reef fishes. For the longest time, I kind of stayed away or shied away from this debate, mostly because I'm not a confrontational person, and it kind of found myself caught in the middle of this, and it's kind of an interesting situation to be in. More recently, a colleague, Andrew Espa, has kind of proposed that we should kind of look beyond just these handful of studies, because there is a lot of evidence, particularly physiological evidence, to show that, okay, perhaps the effects are not as large as initially thought, but we can't just ignore that potentially there could be effects of ocean acidification on sensory systems. Now, more recently, we started to chime in, and kind of our way to approach this debate was to write a commentary. So this was really nice to write this commentary with my trainees, but we kind of are advocating for best practices for performing these olfactory behavioral assays on aquatic animals. And this is particularly because we found that a lot of physiologists have started doing behavior experiments, but we often take a different approach on with how we design our experiments. For example, we tend to starve our animals before performing any sort of physiological measurements. Well, when you're doing behavioral assay, starving an animal will actually make it hungry, and therefore if you're exposing it to something like a predator cue, it's less likely that it's going to be responding to that predator cue if it's actually starving. So the motivation there changes with kind of simple things that we are doing as physiologists, and we need to be aware of these things when we're performing these experimental assays, looking at behavior. So we've kind of shared sort of our experience and our practices around the behavior experiments. I found it difficult to do some of these experiments to begin with, and I felt like I've learned a lot, and we had something to share with the community. So if you're interested, please have a look at it. Now, I'm just going to go back to the physiology. I'm going to let you make your mind up for yourself, but we've done a lot of, we have a lot of physiological evidence to show how ocean acidification and other stressors affecting olfaction in aquatic animals. So this is just showing, or maybe a reminder or a view that the olfactory epithelium of fish is really organized in a very similar way to the olfactory epithelium of vertebrates in general. You have different types of sensory neurons in here in what is known as the olfactory epithelium or the rosette. The axons of these neurons then make up the olfactory nerve. And as these axons travel into the olfactory bulb, this is the part of the brain here where olfactory information is integrated and relates to higher brain centers for a response to take place. As this happens, these axons get organized into different glomeruli depending on the type of receptors that they're coming from. Now, I just wanted to present you the anatomy, just because I'll be talking about responses of the different parts of the olfactory system, including the behavior part. So just to orient everyone. Now, this is work from my postdoc. I'll start with that work because it was kind of the first work that showed that there was physiological evidence for the effects of ocean acidification. And I'll talk about some more current work that we're doing in my lab right now. What I was doing here was using nerve recording from the olfactory nerve of European sea bass. We were keeping the fish in control seawater, so they were not experiencing any hypercapnia or high CO2 in their blood. We were simply changing the CO2 and pH of the water running over their nose and recording the response here to different odorants, shown here by the black bar, added to either control or high CO2 water, and we would randomize the order. This is showing the raw nerve response in addition to the odorant, and this is known as the integrated or the summed amplitude of the response here, and this is what I'll be presenting in the next couple of graphs. So, now, when I was doing these experiments, I tested 10 different odorants. Some were amino acids, as indicated as their important cue for presence of food in the environment. And this is the response to one of those amino acids, L-glutamate. This is the concentration on the x-axis, just to say that these are 10-time increases in concentration, so it's a logarithmic graph, and simply the response to control and under high CO2 levels. So under ocean acidification, as you can see here, there's a decrease in the amplitude of the response to L-glutamate. But what's also interesting is that the detection threshold also increased by up to 5 times under high CO2. So the amount of odorant that was needed to elicit a response above baseline was 5 times higher than in control conditions. So this means that the odorant had to be 5 times more concentrated for the fish to detect it. Now, I also want to show you the response to an a bile acid rather. This is simnol sulfate, is the main bile acid found in chondrichthyans, so sharks and rays. It's used in, actually, bile acids are just like in us, they're used in digesting fats and lipids, but they're also passed through the digestive system and into the water and then act as an odorant. So this would be an important odorant for fish to kind of know that there is a shark or a predator around. And you can see here that again, there was a decrease in the amplitude in the olfactory nerve response to this odorant. Now, we started to wonder what was happening is why is ocean acidification giving rise to this decrease in the response? So some of it is that ocean acidification is likely affecting the molecules themselves. This is work done by a collaborator, Christina Rogatz. She looked at these odorant peptides that are involved in mediating egg ventilation in female green shore crabs. And what she's showing here is that the odorants become more positively charged under a low pH environment, which changes the charge distribution around the molecules themselves. So this means that these odorants have a harder time binding to the receptors because of electrostatic interactions are really important part of how odorants bind to their receptors. Now, we also think that these charges are also changing on the receptor side of things. So therefore, adding to this impairment of how the odorant kind of binds to the receptor. There's still more work to do on that space, but basically that's part of how we think that there's a decrease in olfactory sensitivity in aquatic animals. Now, I wanted to see all of these experiments, initial experiments were acute. I wanted to see if there's any sort of indication that the fish might be compensating for this decrease in their sensitivity in the sense of smell. So I did transcriptomics. So what I did was expose juvenile European sea bass to ocean acidification for 2 and 7 days. And I took out the olfactory epithelium and olfactory bulb, so 2 different parts of the olfactory system, and looked at changes in gene expression at 2 different time points. These are quite short, but I wanted to capture what was happening as these fish were exposed to ocean acidification. So we found some really interesting results. There were genes that were downregulated in the olfactory epithelium, such as olfactory receptors, those involved in maintaining cell excitability. Not a good thing for a neuron to decrease its excitability in response to not being sensitive enough. And again, decrease in genes involved in neuronal growth and development. And these are juvenile fish, so their olfactory fish system it would be developing at this time. Now, in terms of the olfactory bulb, we saw genes that indicated that there's decreased synaptic strength and decreased cell excitability. So all this to say that, in fact, they weren't compensating, but doing the opposite. Their olfactory system seemed to be becoming even less responsive over those next few days. So there was no compensation. It was actually kind of turning down the light, not using their olfactory system kind of meant that these olfactory neurons are becoming less sensitive and less used in response to ocean acidification. So this was a little bit alarming, well, a lot alarming, but we also wanted to see if this would then kind of translate to the whole animal and changes in their behavior and how they detected a predator smell. And this is just showing a change in activity of these fish after 2 days of exposure to ocean acidification. See control fish reduce their activity, a normal response to a predator cue, and then realizing that there's just the cue there, that the predator's not there, they kind of come back and regain normal activity. In control fish, what I was seeing is that the fish responded to a lesser degree. And this was a statistically significant difference. I've tested these different fish at 7 and 14 days. And overall, what I was finding is that elevated CO2, exposure to elevated CO2 caused up to a 40% decrease in baseline activity, up to 66% decrease in response to predator cue, and up to a 150% increase in freezing behavior. I'm not showing this those data right here, but this is consistent with some of a work from other researchers showing that they're a little bit more skittish after exposure to high CO2, and there's some reasons behind that, that it might have to do with increase in anxiety. So overall, this was translating into changes in the whole animal behavior. So what we did at the with all this information was we proposed a novel physiological mechanism for how ocean acidification affects olfaction in fish. And that is that changes in these odorants give rise to a reduced binding of odorants to receptors, which gives rise to a decreased response and or detection, as shown by electrophysiology. We show that there's a decreased excitability of receptor cells and olfactory neurons, as shown through our RNA-seq data, as well as decreased synaptic plasticity and strength, indicated also by our RNA-seq data. And this was all kind of translated into impaired behavior and potentially learning about these odorants at the whole animal level. Now, when I started my lab here at UTSC, that was about 4.5 years ago, we started to wonder, are other animals affected by ocean acidification in a similar way? Particularly, a lot of the previous work was done in fishes, and we started to look at other animals such as invertebrates. So we studied these amazing crabs. They're Dungeness crabs. I really kind of was a fish person before, but really fell in love with working with the crabs. I just want to tell you a little bit of how olfaction works in these animals. You can see here their antennules, the little things that are flicking down. Those are their antennules, that and the tops, the bristles there, the acetacs, are where the olfactory sensory neurons and their dendrites are found. The cell bodies are found just beneath here, the cuticle. Sorry, I'm trying to move it with the left hand here. So the sensory neuron bodies are found here, and then the axons are projecting through the antennule and into their olfactory bulb and higher brain centers. So their flicking behavior is more like sniffing in dogs. So they're increasing their flicking when they're trying to detect an odorant to increase the chance of odorants ending up on the bristles in order to detect them. So as I mentioned, we were studying Dungeness crabs. These are crabs that form the most valuable fishery in the Pacific region in Canada. It's worth over $250 million. They're found all the way the Pacific coast, so the other side from you guys, on the west coast of US and Canada. And this is just simply showing their life cycle. I'm not going to go into it, other to say that we were studying the adults, but there had been one previous studies done on the larvae of these crabs, and this is looking at how upwelling events during the spring, during coastal upwelling events are affecting the larvae of these species. So you can see here that CO2 levels can get up to 1,300 microatmospheres, so even higher than what is predicted for the open ocean for the end of the century. And this study is showing that exposure of these larvae to these levels of CO2 can affect their carapace. Basically, they have trouble forming their carapace due to a decrease in carbonate, which is necessary for carapace formation. due to ocean acidification. And they're also showing that the mechanoreceptors found on the bodies of these larvae are also missing or damaged. So they have less ability to kind of tell their orientation in the water, because of these damaged mechanoreceptors. So really, this was the only study that was done before we started our work. We decided to work on the adults because, and we only studied the males because we're not able to collect or harvest females because just general regulations around keeping the population viable. So we, just like we did in fish, this is Dr. Andrea Durant, my former postdoc, who was doing the electrophysiology in the crabs. So what she did was remove an antennule. She would keep it in a dish and perform electrophysiology nerve recording from the antennular nerve, where she was adding and changing the solutions of the water going over the estatax, or the sensory neurons. And just like I did, she would add different odorants to different water chemistry. The difference here was that these crabs were exposed to ocean acidification for 10 days, and we can compare it to their response to control crabs who weren't exposed to ocean acidification. So what I'm showing you here, again, is this integrated nerve response in response to a odorant, in this case, cadaverin. And cadaverin is a food cue for crabs because they are detritivores, whereas it really is smells like, it sounds like it. The smell of rotting flesh is what cadaverin smells like, but to a crab this is it smells delicious. And just to show you that the response of these crabs under high CO2, or those exposed to high CO2, was much lower than control crabs. We also tried to find a deterrent, or looked at the response to a deterrent, which is ammonium chloride. This is just a source of ammonia would indicate to another crab that they might be frightened. Another crab might be frightened and releasing ammonia during perhaps a predator response. And we saw similarly that the integrated nerve response was decreased to ammonia when they're exposed to high CO2. Now, we also collected antennules, the other antennule from the crabs. And what Andrea did was perform immunohistochemistry. We had an antibody that was able to detect a protein that's involved in olfaction in crabs. It's an ionotropic channel involved in olfaction in crabs, and she double labeled with DAPI to show the cell nuclei here. And you can see these grape-like clusters are the clusters of the cell bodies of the olfactory sensory neurons just below the cuticle. So she compared crabs that were exposed to high CO2, and they had fewer cell fluorescence, therefore indicating that they're expressing fewer of the IR25A receptors in the olfactory sensory neurons. And what was interesting was that these cells were also smaller in surface area, and we calculated that in volume as well by up to 26% to show that these neurons were in fact undergoing hypertrophy after exposure to ocean acidification. Again, I think this is going back to this concept of use it or lose it. Neurons take a lot of energy to maintain, and we think that this is kind of related a way that these animals are perhaps saving energy by downregulating cellular processes perhaps that are happening in response to ocean acidification. And because the whole pathway is seen as not being used as much, and therefore why maintain something you're not using, right? It's similar to our muscles that would go under hypertrophy. Now, at the time, I also had a wonderful undergraduate student, Elisa Kodikian, and this is her work that she was doing during her undergrad. She then stayed on to do a master's, and I'll tell you a little bit about her master works in just a minute. But we also wanted to look at their behavior. So again, these crabs were exposed to control and high CO2 conditions for about 10 days. And what she did was had these crabs in a tank, and she let them kind of overcome handling stress for about 30 minutes, and then she would add progressively higher concentration of cadaverine into the tank and looked at their antennular flicking behavior. At the end, she did a pulse of ammonia chloride to look to see how they responded to this deterrent. So the data are a little bit complicated, so I just wanted to show you the control crabs and how they were responding. So this is at 0 odorant concentration. We had this baseline flicking behavior, and then they started increasing their flicking behavior at 10 to the negative 6 molar of the odorant concentration, which was cadaverine. Again, at the slightly higher concentration, they were increasing flicking behavior, but at a very high concentration of 10 to the negative 4 molar, they started inhibiting the flicking behavior. And this makes sense because there's enough odorant in the water that they don't need to increase flicking behavior, or they're sniffing. Now, in comparison to the crabs exposed to high CO2, what was interesting is that they increased their antennular flicking only when the concentration was 10 to the negative 5 molar compared to 10 to the negative 6 molar in the control crabs. So here the cadaverine had to be 10 times more concentrated before they increased their response to deodorant. So this all indicates that they have a decrease in sensitivity in their response to this odorant. So just a little bit of recap the evidence so far that I've shown you. I've shown you that ocean acidification affects the sensory neurons themselves. I've shown you evidence from the olfactory bulb, and I haven't shown you, but this is another group working on this where they're looking at how ocean acidification affects central processing in terms of changes in neurotransmitter functioning in the central nervous system. As well as all of this is what we do. We kind of like to look at multiple level of biological organization and kind of bring it all back to the whole animal responses with changes in behavior. Now, what we're working toward is this unifying theory for how ocean acidification impairs olfaction in aquatic animals. So basically, the high CO2 or low pH is affecting and causing changes in odorant, and particularly in the distribution of charges around the odorant, likely happening around the receptor as well, which is impairing the ability to detect deodorant, and therefore changes the receptor odorant binding affinity. This is kind of leading to a reduced detection and response of these odorants by the sensory neurons and even hypertrophy. This can have either direct effects altering behavior if these odorants are not detected, or there could be longer term central nervous effects that are then further impairing the response to these odorants. Now, when we work on this, a lot of our reviewers, and I get a lot of questions kind of around this topic. So we started to look at how animals in coastal environments, are they better or more resilient at responding to high CO2? So what I'm showing here is a figure from Bauman a review by Hannes Baumann that was published in 2019. And this idea that because the open ocean has fewer fluctuation in CO2, so it's a more stable environment, therefore these animals living in the open ocean should be more sensitive to CO2 than the animals living on the coast. But what I've shown you so far is evidence from European sea bass that live on the coastal shelf, and the crab, the Dungeness crab, which live on the coastal shore. So we wanted to see, look at an example of an animal that lives in the near shore to see if this is true for those animals as well. So this is where Elisa stayed to do a master's in my lab, and she was looking at the Hemigrapsis oregonensis, or yellow shore crab, which is living in the near shore and actually in the tide pools near the Pacific. And this is work that we did at Banfield Marine Sciences Center. Now, I'm gonna show you some of the effects for these nearshore crabs. These are acute effects of ocean acidification. So we simply put the crabs into control conditions. We look at how they respond to an odorant, again, a food odorant. They prefer it. We then change and move them into high CO2. They actually are confused. They tend to not like it or respond to it very much, and then we put them back in control conditions and they're responding to the odorant again. So really acute effects in what is supposed to be a species that lives in an environment that encounters high CO2 on a regular basis. Now, we also are showing here that they're slower to detect deodorant. So they, when we measure this in the time taken to get to where the deodorant was found, which was on a filter paper, and they're 3 times as slow at detecting the deodorant when they were in high CO2. And there seems to be even a lingering effect here, because when we put them back in control water, they're still slower to detect deodorant in high CO2. So it's an interesting, we think it's a bit of an effect that kind of lasts perhaps beyond the half an hour of being back in controlled seawater. But it's absolutely reversible, which is interesting. We also did, or I say we, but it was Elisa. She performed experiments and exposed crabs for 2 weeks to control kind of medium levels of ocean acidification, akin to what they're experiencing during an upwelling event, and then also future predicted high coastal levels of high CO2, which were about 3,000 microatmospheres. And what she was doing was looking at the behavior of the crab. So first, she was measuring their baseline, just letting them get over handling stress. She temporarily caged them and added a food cue, putrescine in this case, to a filter paper, and this was all randomized. And then we looked at the behavior of the crabs. So what I'm showing you here is their odorant detection. So just like, so what we saw, actually, we looked at the distance traveled, how much they moved around during this time, and we found no significant difference in the total distance traveled. But again, just like I showed you in the acute experiments, these crabs took about 3 times longer to detect the food cue when they're exposed to ocean acidification for 2 weeks. So again, this there's no sign of that these crabs are compensating in any way in terms of their response to an odorant. Now, I wanted to also show you the preference for the odorant. So blue show controls, whereas yellow is medium, and orange is high CO2, and you can see here there's a significant difference in their preference for deodorants. And this is showing during the first, second, third, fourth, and fifth minute of exposure to deodorant. And again, just like in other animals, after 4 or 5 minutes, they get bored. They realize the food maybe is not there, and they kind of go back to their normal behavior. But this also indicates that there's some kind of an olfactory impairment taking place during exposure to ocean acidification. Okay, so the last question we had was, well, what about, these are static exposures. What if they're exposed to a fluctuating CO2 level? So the levels fluctuated between about 700 and 3,000 micro atmospheres to kind of mimic that changing environment, and the average was around 1,500. The average CO2 they were exposed to was around 1,500 atmosphere, which is our medium control. And these crabs were held at 1,500 microatmospheres the whole time. And this time we did 1 week long exposures, followed by the same behavioral testing. This time we also did some immunohistochemistry, so I want to show you those data as well. What are we doing for time? I'm going to try and wrap up in about 5 minutes or so. So here are the behavioral data and how these crabs were responding to these fluctuating and static conditions. I'm just going to highlight, Lisa also looked at how they recovered, but we're not going to focus on that. It's just a little bit too much for this talk to cover. So this is the percent of time spent on the odorant side. Again, they're preferring the food odorant when they're exposed to medium CO2. They showed less preference. And then when they're exposed to fluctuating conditions, their response was sort of intermediate. This is also shown in the time to detect odorant. Again, they had a longer time to detect deodorant, and they're significantly different than controls, whereas the fluctuating group was not significantly different than either medium and control groups, indicating that they were somewhere in between in terms of their response. And this is just showing the IR25 labeling. Elisa also did that after these crabs were exposed to either control, medium, or fluctuating CO2. What was interesting here is that these receptors were, there's significantly less of them under medium conditions, and again with fluctuating conditions they were not significantly different than controls. So indicating that perhaps when they're exposed to fluctuating conditions, they have this time when they're in normal CO2 to kind of recover their response or give them a break from the high CO2, and therefore this is also seen in fluctuating responses to hypoxia, for example, not necessarily to odorants, but to kind of respiratory physiological responses. We used BRDU, so this is an indicator of cell proliferation. We wanted to see that whether the medium group here, did they have a smaller cell size or less fluorescence because these cells were actively dividing. We didn't see a significant difference between the amount of proliferation in these cells, although maybe perhaps there was a trend here for the medium group, but this wasn't significantly different. So it's not that these cells are proliferating more when these crabs are exposed to high CO2. Now, just to answer this question, are the coastal species more resilient to ocean acidification than open ocean species? Well, at least the one species we looked at doesn't seem to be, and this kind of prediction doesn't really fit the trend of what we're seeing in some of the nearshore animals. Of course, we've only looked at one species, so there's lots more to to look at and compare. But for now, we don't have any evidence really to show that they're more resilient to ocean acidification than those that live closer to the open ocean. Now, just a little bit, I want to get into what we're currently doing and some future directions. This was a study done by Berger et al. That was published in 2021, and these are on, the study was on Dungeness crabs and really predicting the vulnerability of the species to these three different stressors, or, deadly trio stressors. They were predicting the present vulnerability and future vulnerability of the species, and it was weighed by the life stage. Then they predicted that their vulnerability is going to increase in response to pH, dissolved oxygen, and temperature in the future. So they're already vulnerable now, but they're going to become even more vulnerable from these other stressors. So work in my lab was just recently started looking at how hypoxia affects olfaction in marine animals. My PhD student and I did a review, and we were surprised to find no studies looking at how hypoxia affects any aquatic animals. So we've just recently published this earlier this year, so just wanted to show you a snippet of this work. It's just a short communication, but we're excited to show these new findings. So does environmental hypoxia affect olfaction in fish? The study was done on gilt-head sea bream, and Liam traveled to Portugal to do this work in collaboration with Dr. Peter Hubbard. They did electroolfactograms, so measuring again the nerve or the olfactory response of these fish under mild hypoxia. So this was only 50% saturation, and moderate hypoxia, which was about 30% saturation. Again, and this is the response shown to L-cysteine, an amino acid. And what we're showing here, that there's a to these odorants. So very exciting, and the first look at showing that mild hypoxia could be affecting olfaction in aquatic vertebrates. Okay, and then just the last couple of slides, more future plans. We have an aquatic facility which has been newly renovated. We're about to kind of get in there in the next couple of weeks. We have some, it's really done to a high spec. We have some really cool multi-stressors that we're going to start to look at multi-stressor and multi-generational studies, so longer term exposures to see, to look more into these questions of are these aquatic animals able to adapt to this rapid change in their environment, and how do these stressors then combine to affect their olfactory system. I just wanted to also say that kind of bring this to kind of a wider perspective, I guess. This is where the fish that we consume, and as people, so about half of the fish that we consume comes from wild capture fisheries, and this level has leveled off since new fisheries regulations were introduced in the 1980s, preventing us from overfishing. And in the meantime, aquaculture's kind of taken up the slack in order to kind of keep up with the demand of a fish as a protein source, as it's an important and the main protein source for many people around the world. However, aquaculture itself is not immune to the CO2 problem. What happens in aquaculture recirculated systems is that fish produce carbon dioxide as a product of a metabolism. CO2 is 20 times more soluble in water than oxygen. So it really stays in there and it's hard to remove. And when you're circulating the water, you're basically accumulating a lot of CO2 in the water. And this gets to such high levels that it starts to affect the growth and physiology.
I think we're experiencing some technical issues.
Okay, and sorry, this was a little bit long. I just wanted to leave you with this quote for Dr. Robin Wall-Kimmerer. She's the author of Braiding Sweetgrass. But again, as I alluded to at the beginning of the talk, is if we really kind of lived by some of these teachings from indigenous cultures, we probably wouldn't be in the climate change trouble we are right now. So I really like this of, never take the first, whether it's animal or plant, never take the last, take only what you need, take only that which is given, never take more than half, leave some for others, harvest in a way to minimize harm, use it respectfully, never waste what you have taken, share, give thanks for you have been given, give a gift in reciprocity for what you have taken, and sustain the ones who sustain you, and the earth will last forever. Final acknowledgements to all my wonderful trainees, my funding sources, NSERC and CFI, and BMSC for hosting us for experiments there. Thank you.
Thank you so much, Dr. Porteus. Sorry, I think you cut out for a couple of seconds there. So if you ended up stopping sharing your hello?
I'll stop sharing. Hello?
Hello? Can you hear me?
I can't hear you, Deepal.
Oh.
Hello?
Hello, can you hear me now?
I'm not sure what's happening, but I can't hear anyone.
Okay. If anybody has questions, please feel free to type it in the chat. If sure why.
Hello?
Hi, can you hear me?
Oh my goodness. Yes, I can't hear you. I'm so sorry. Let me see.
That's all good.
Was I sharing the screen or was I not sharing? I'm just. It cut out at some point. Oh my goodness. When did this happen? Oh, okay. I was like, oh no, did I do the whole talk? Let me see what's happening with the sound. I'm not sure why I can't hear you guys. Hello?
Hi.
Oh, okay. Hi, now I can hear you. I'm not
Perfect. Okay, that's all good. Yeah, you ended up cutting out there for a little bit, and that made you stop sharing the screen.
Okay. Was that during the multi-stressor?
It was the last 2 or 3 slides after the slide of the stressors.
Okay. I'm so sorry. I didn't realize. I'm relieved it only happened at the end.
Yes, no, that's all good.
Okay, so I can just reshare quickly just to show you. So I was just saying that we have a new aquatic. Whoops, am I sharing?
Not right now, no.
Okay. Right, here we go. I was just saying we have new aquatic facilities. We're about to move into them. They've been newly renovated, and we are we have some really nice multi-stressors, and we're going to start looking at multi-stressor, multi-generational studies in kind of this space. And then I just had quickly, I was just talking about wild capture fisheries and aquaculture, basically where our protein comes from. About 50% of it still comes from wild capture fisheries, but aquaculture has its own CO2 problem, as CO2 tends to accumulate under high CO2 conditions because it's a product of metabolism, and these in recirculated water, CO2 just stays in there. And creates problem to, it gets to such high levels that it can affect growth and immune response of fish. And we're kind of looking at that using lake white fish currently. And just wanted to leave everyone with a quote from Dr. Robin Wilkemmer about, you know, this is kind of a way that Indigenous cultures look at the environment and how they interact with anything, any animal or any plant. And it was just, I think it's a really nice quote from this book. And just wanted to acknowledge my funding sources and my wonderful trainees and collaborators. But I'll leave you with that maybe while I answer.
Perfect. Thank you so much. So are there any questions for Dr. Porteus here? I guess I'll give it a minute or two to type it in. In the meantime, though, thank you so much for the awesome talk, Dr. Porteus. It was very interesting. And thank you all for joining us virtually. Here in Newfoundland, we are currently in the middle of a snowstorm. So thank you all for
Yeah,
joining us.
and I can appreciate that. And thank you. And I understand there's not many people attending. We had 2 snowstorms in the past week here. So I
Oh
can,
my.
I can relate.
Yes, it always happens at the worst time.
Yes. Hope your animals are safe and they're being looked after, because that was our biggest challenge.
Mhm. It seems like there are no questions. And once again, somebody asked if this seminar is recorded. It will be, and uploaded to the MUN BGSA YouTube channel. Link is in the chat there. Anyway, thank you all so much for coming, and thank you once again, Dr. Porteus, for your
Thank you, Deepak.
seminar.
Thank you, everyone.
See ya.